also recording for that cannot take the class, so this is going to be recorded. So now today's topic is about mitosis and the cell cycle, basically. And a lot of you might be aware of what it actually is, but let's see what is needed today. So now the learning outcomes given in your syllabus include a few of the things like understanding a chromosome, understanding what all of these structures are, understanding what mitosis is for, understanding mitosis itself, and understanding the minute details of mitosis. So I'm assuming up till here we'll be able to complete mitosis, and the rest of they're very short, but we'll be doing some of them tomorrow because I don't want to overpower you with all of that information today. So let's start from something very basic. Now, before jumping on to reading the text or looking at the diagram, I want you to move to this picture right here. So this is a very generic picture. It shows you the nucleus. Why does it show you the nucleus? Because I want to make sure that you know when we're talking about DNA, chromatin, chromosome, whatever that thing is, I want you to know that everything is happening within this nucleus, right? And then within this nucleus, so since we are talking about nucleus, within this nucleus, there are two structures. You can see these thin lines, and then you can see this thick ball. These thin lines are actually, in fact, all of that DNA that will get replicated or that will be used to make proteins very easily, whereas this thick ball cannot be used easily. Now, for now, this point isn't as important, but it's important to know it for the next topic, which is DNA replication. But one thing I want you to note from this point here: whenever the DNA is packed thickly, whenever it is packed so thick that it's not replicating, all it's doing is existing and being packed and stored at a side, it can be stained. The nucleolus can be stained, which means when we are replicating cells and we look at things like this that we call chromosomes, they are in fact looking like this because they are highly packed. And when we come to the word chromosome, then I want you to understand all of these terms separately. I want you to understand the difference between DNA, the difference between chromatin, the difference between chromatid, and the difference between chromosome. Now, what is DNA? DNA is just a polymer made out of many of those nucleotide monomers. Look at this part of the diagram, a double helix of a DNA. Because it cannot exist freely in nature, because it can easily be broken down and it's so big that it can stretch up to the moon, we need it packed tightly. So how do we pack it together? And only when it's packed tightly can it be stained to give a separate color and visible by a light microscope. So how do you pack this DNA? You pack it, and let me just tell you here, even if you don't know, DNA is slightly, its polarity is negatively charged or is it positive? So I think it's positively charged. I'll have to go back and confirm that. Yeah, and I think histone proteins. So what DNA is packed like is DNA is wound up on beads. Can you appreciate these small beads? These are called histone proteins, and they're positively charged. So what happens is there's this charge attraction that leads to this DNA binding first along this blob of histone proteins. This blob is made out of 8 histone proteins, as you can see here. When these blobs form, DNA is slightly packed. And this packing, when multiple of these DNA wound upon histones pack upon each other, they form a chromatin fiber. A chromatin fiber is a packed form of DNA. And when so many of these chromatin fibers wind up around each other, they can form a chromatid or a chromosome. Now, what's the difference between a chromatid and a chromosome? When you have this single structure, it's a chromatid. When this single structure, for instance, this obviously had DNA in it, when this DNA replicates, it's obviously going to become a double structure, right? This is going to make an exact copy of itself right here. So this replicated copy is what forms the chromosome. Can you see? Can you appreciate this chromosome and this one, how similar they are? So one of the limbs, let's say this limb is a chromatid. And when it replicates, it makes an exact copy of itself. It forms a chromosome. And now we will see why it needs to make this exact copy of itself. Okay. So again, coming back to how your book defines it, your book says a chromatin is made up of, it's the material of which chromosomes are made. Okay, we know that chromosomes are the final product. So you see, you don't need to cram all of these terms. You need to be able to understand what's happening, right? It's basically just DNA and proteins, these histone proteins and other stuff wrapped around each other. Again, what is chromosome? Chromosome is tightly coiled chromatin, just like we talked about. Now, it says it's visible during cell division. I just told you, highly packed DNA, just like this one, is DNA is only visible when it is highly packed. And so, what happens in cell divisions? In cell division, because DNA is no longer being used functionally to make copies or proteins or RNAs or whatever, it becomes tightly packed. And so, when it is tightly packed, of course, it is visible. Now, its tight pack formation is chromosome. So, in cell division, what you're seeing, the chromosomes that you're seeing, is in fact DNA tightly packed. Okay, and then we know that in prokaryotic cells, the DNA is not linear, it is circular, it is a full circle because it doesn't have a nucleus, it's self-protective and circular. Again, we know nucleolus and the other ones are very basic. Okay, so let's move on and see. What is this called? It's called a karyogram. What it's doing basically is it's showing all of your chromosomes. Now, what I want you to appreciate is the first thing, this is just tightly packed DNA. It's a lot of tightly packed chromatins. When they pack together, they form these structures. These structures we know this DNA has been replicated once. How do I know that? Because this is not in the form of chromatids. All of the chromosomes, all of the structures I see are double formed. They are not single form, which means they have replicated once. And that is true because when it has replicated, then it is ready for division. And when it is ready for division, it is tightly packed, and that is the point when I can see it. Otherwise, all I will see is a nuclear envelope and a small nucleolus because all of the other DNA will be open. So I hope that point makes sense. The other thing that you need to note is that not all of them are similar. Some have a long limb, some have a short limb, with some, and you can see this center of attachment between this chromatid and this chromatid. So there's specific names for these that we need to talk about, and those names are gonna come up. And can you appreciate some of them have very small upper tails, some of them had longer upper tails and smaller lower tails. So let's see what all of these things are called. So let's start over here. Again, let's appreciate that a chromatid or a singular chromosome, this is the one, when it replicates, it becomes this butterfly shaped. Look at this, it says sister chromatids. Why does it say sister chromatids? Because this chromatid resembles this chromatid because it has exactly replicated. The DNA has exactly replicated. And remember I said there was this little attachment in the center between this arm and this arm, the long arm and the short arm, or the short arm, which is called a P arm, and a long arm, which is called a Q arm. The attachment between them, or the attachment between sister chromatids, both of it is called a centromere. Okay, as simple as that. Okay, the next point, now coming to this diagram right here. And this one just shows you how they look apart. It's again showing you chromatid, and then how they can be equidistant, they can be long and shorter and taller arms. Now, the next thing you need to understand, and here again, you can look at the centromere. It is a non-coding region, which means, so what's a coding or non-coding? So let me tell you here, you haven't done DNA transcription, translation, anything right now. So just let me just tell you this right here. What are genes? When we say gene, what we mean is that in our DNA, in a piece of DNA, if this is my piece of DNA, a small segment in my piece of DNA, for instance, this segment, that codes for a particular protein chain. Please focus on my words. Make sure you understand what I'm saying. Look at this, appreciate this protein chain. It is a small segment on the DNA. For instance, look at this green segment. This green segment on this DNA is a gene coding for a specific protein. It's different from yellow. It's different from this pale color right here. It's green and but it's similar. This gene is similar to this gene right in front of it on its sister chromatid. Why is it similar to this gene? We know why, because these are sister chromatids. They're just replications of each other. So this must not be such a difficult question to answer mentally if you understand that this is one chromatid and this is a replicated form, its own replicated form. Then you understand that the location of a gene, for instance, I say the gene for being tall is this red one located right here. Then if your DNA replicates and forms this chromosome, then the gene of being tall is always going to be the one right opposite to it on the other strand, because this is an exact replica. And then genes, and then there are areas within this chromosome that are non-coding. And there are a lot of areas like that, but two specific ones that you need to know right now are the centromeres. These are areas that have no coding. They're just there for strong, for tensile strengths, right? And then there is something called telomeres. They're both of the edges of the chromosomes. Look at this one, the last pieces. Now, these telomeres are also non-coding and they have their own specific function that we will talk about very soon. But for now, just understand this much. Just understand the basics of chromosome. So is this clear? Any questions up till now?
No,
Teacher.
no, teacher.
Okay, perfect. I hope if there were any confusions, they've been cleared by now, okay? I hope that's happening. If it isn't, just ask me again. Okay, now what is this staining? Now, this is a special staining, and we call these immunohistochemistries because we stain them under certain light. And what they're highlighting is, remember those telomeres that were at the end of each chromosome? So let me just make out one chromosome for you here. And that's the fun part about these immunohistochemistry diagrams, because they're so specific. So look at this one. These, if I say these are the telomeres because they're at the edges, then this is one chromatid, this is the centromere, and this is the other chromatid. Now, will you guys be able to make this diagram for each one of these chromosomes? You will, I'm sure, because it becomes very easy if you understand these telomeres. And the specificity of this diagram is that they've stained telomeres specifically to show you that every dividing cell will have telomeres. Remember this, every cell that is in senescence, that wants to divide and live, is supposed to have telomeres. If there are no telomeres, cells do not divide, and their eventuality is just death. Okay. So let's see now, moving on to the basic topic, the cell types, the cell cycle, the horrendous cell cycle that everybody is always so scared of. It's a very easy cycle. If you understand it and you just go with it. So just don't read anything on the slide yet. Let's move with my pointer. Okay, let's see how we go that way. So I'm just gonna change my pointer color to, let's say, okay, let's go with purple because I haven't used purple in a while. Now, okay, let's use a highlighter instead. Okay, let's see how this goes. So I want you to remember that a cell cycle, what is a cell cycle? It's basically, like it says, a cycle. So it's supposed to have a starting point and an ending point. In a cell, it is basically the sequence of the process in which a cell is born and then it grows and then it comes to a point that it divides to form another version of itself. So now that one cell will become two cells, and that is the cell cycle. And because it's forming a new cell, which is why we call it a cycle. In a cell cycle, what you initially have is one cell, and this one cell will just divide to give us two cells. And these two cells will be exactly identical. Even their DNA will be identical. Remember those those chromosomes. So just imagine that if you're this cell, just imagine if you're this cell, you're clever enough, if you had this set of chromosomes and you're clever enough to give one of this set here and one of this set here. So you give one chromatid here and one chromatid here and you become two cells. And at any point in the cycle, each of these can make their own new chromatids from the one chromatid they have to give two cells of their own, right? These new cells with new chromatids that were exactly identical to this one. So in fact, it is a cycle. Now, what we need to understand now, what do we need to do at this point is realize that we have to divide it into three phases. There are exactly three phases. The first phase is interphase. The second phase is mitosis, and the third one is cytokinesis. So again, first phase, interphase. Let's ignore everything else for now. Interphase, mitosis, and cytokinesis. Now, all we're going to do is go about what the words say. So mitosis, let me tell you, means that one cell is going to divide into two, just like I told you. One cell dividing into two is mitosis. Now, the whole process of mitosis isn't this. This whole process that I told you isn't exactly mitosis. The process that we've mentally associated with mitosis isn't this entire division. This entire division is, in fact, the cell cycle. Then, what is mitosis? Is, in fact, simply that if this is one cell and this is its DNA, its nucleus, mitosis is division of the nucleus into two equal parts. It's simply this division of the nucleus. And because nucleus is the life of the cell, if there was no nucleus, no cell, errors in the nucleus, different diseases. So mitosis is this division of nucleus. The second process, the cytokinesis, as the word says, cyto means cell cytoplasm, and kinesis means energy, breakdown, reaction. Cytokinesis is the division of this cell. So mitosis is division of this nucleus, cytokinesis is the division of this cell. Then what is interphase? Interphase is everything that was before and everything that will be after. What's that supposed to mean? It means that these two baby cells, when they become baby cells, they have to grow bigger and bigger. And so from this point onwards, from the point of cytokinesis, as these cells continue to grow to finally become this large cell that it has the capability for its nucleus to divide, all of this process is just the interphase. So if you understand this, you understand that mitosis is division of the nucleus, cytokinesis is division of the cell, and interphase is the whole growth phase. It's the whole teenage adulthood till the cell, let's say, gets married and has kids. So if these little ones were its kids, it's not a good analogy, but just to make it easier. So that's simply it. Now let's see how we go about it. Okay. So we say, let's start from interphase then. Let's say if I gave you a baby cell, I'm starting from here. This is my start line. I gave you a baby cell. Now, all of these cell cycles are divided at this is my start line, and this will be my start line will in fact be my finish line because it is a cycle. Okay, so now interphase, mitosis, and cytokinesis. Out of these, the interphase and mitosis are divided into phases, right? For instance, for the interphase, we have three phases. We have something called a G1 phase, something called an S phase, and a G2 phase. Now, simply putting it here, G1 and G2 have relatively similar functions. They're called growth 1 and growth 2. Simple, very simple, similar functions. Then what is the difference? What is this S phase that is so different than these G phases? So I'll tell you now what's the real difference. I told you if G stands for growth, so G1 is growth 1 phase, G2 is growth 2 phase. What happens in between them that we have to separate growth 1 phase from growth G2 phase. What happens, in fact, is that in growth between these, let's assume we had a chromatid over here, or we had a chromatid with a single DNA over here. In the S phase, DNA replication takes place. Chromatid now becomes that chromosome that is soon going to be visible as soon as I stain the cell, which means now we have two copies of DNA available, not just one, but two identical copies. So G1 is the growth phase for the cell when there was a chromatid and one copy of DNA. GS phase came in where replication took place, and then G2 phase came in where now this large cell is again going through a growth phase. As simple as that. Okay, now let's see what each of these individual phases account for. So we say G1 phase is a growth phase, which means cell is metabolically active, its organelles are replicating, its cytosolic components are replicating, it is thriving, more protein is being produced until the point it finally grows up into the point that it says, okay, now I'm a grown-up cell, I'm gonna enter S phase, in which my DNA, the moment its DNA starts replicating, it comes in its S phase. And not only the DNA, but the cell, every cell that replicates its DNA does not do it to just waste that energy. It does it because it wants to divide, because at some point it has decided, now I'm capable, I am going to divide. And what is division essentially? Is division just breakdown of the cell into two? No, I told you, breakdown of the cell into two is in fact just cytokinesis. The cell is breaking into two parts from one. What is important is mitosis, breakdown, perfect breakdown of the nucleus without any errors. And that breakdown of the nucleus is what's so important for us at this point. So every cell that has replicated its DNA is not doing it for cytokinesis or to enter G2 phase. It is doing it for this specific thing called mitosis, because it wants that DNA to be given to a smaller baby that is exactly its identical and performs the same function, does the exact same thing. And then when this big cell, now imagine, now this big cell, when it enters growth 2 phase, so what it needs is, obviously it's going to perform all of the activity that it was performing in the growth 1 phase, but now it has its mind set. It has its mind set towards mitosis. So what it needs to do is it's going to start preparing a little bit for mitosis as well. What does it need for mitosis? I'm sure even if you don't know what mitosis is, you know that if it's a process, it's going to need energy anyways. So for if it's going into mitosis, what it's going to do is start making lots of protein, start making lots of ATP, start collecting lots of nutrients for all of it. So it's again going through a growth phase, but this growth phase is relatively slightly different because now the target of the cell is very different from what it was in the G1 phase. And if I say that the target is different, which means what if a cell decides, for instance, neurons, even if you don't, let me tell you here, neurons, if you cut off a neuron or if a nerve is cut off, what happens? If a person becomes paralyzed for life, do they recover? No, most of the permanent paralysis, if your vertebrae is cut off from your leg below, you lose your leg, even if you stitch that leg back, put on prosthetics, whatever you do, your same neurons cannot grow back, which means those neurons never replicated their DNA. They never had that ability. They were never doing their S phase, their G2 phase, their mitosis phase. They were always in their G1 phase, which tells us that some cells will always be in their G1 phase. So what happens to these cells? Do they always remain small? No, not at all. What happens is, in this slide, one thing that's missing is this phase. Cells that grow big and have, okay, I don't like the purple color anymore. Gotta go back to red. It was this, red is this dark in color. It's very scary, but makes a point out of it. Okay. So in this phase, see, I can actually see it better. Okay. So in this G1 phase, if a cell is not, my writing is so bad. So if in this phase, if a cell is, for instance, it's not going to be stuck here, it's going to grow up and go into something we call the G naught or the G0 phase. And from this G naught phase, it means the cell is mature and it is thriving and it's going to do all of its functions. It's just not going to replicate or go into the S phase. But what if at some point that cell decides, for instance, when you are 40 years old and your skin cell that was in G naught phase decides that, okay, now my skin is bearing a lot of wear and tear, I should replicate and make a new cell. So some cells have the ability to go back from the G naught phase into the G1 phase. Why? Because so that they can enter the S phase again. And so once they enter that, they're going to go into the G2 phase. How is the G2 phase different? It's going to accumulate protein, ATP, lots of content. And from that onwards, from the G2 phase, at any point when the cell replicates its centrioles, remember those organelles called centrioles? I call them the MTOC. Remember the MTOC, the microtubule organizing centers. I told you microtubules. You remember the microtubules, the alpha and beta subunits required for moving things within the cell. I called them the railway tracks of the cell. We all acknowledged that they were used for moving everything within a cell. They're basically internal transport of the cell. And so centrioles, I told you, were the microtubule organizing centers. They decide what the microtubules are doing. So the last point, the last nail in the coffin to start mitosis is this centriole replication. Do you remember centrioles were already these two cylinders at 90 degree to each other? The moment there are two more of these, the cell makes two of these because it was already correcting protein and ATP. It makes these and then it decides, okay, now I am entering mitosis, which is division of the nucleus. Okay. Okay. So let's see. Now within mitosis, there are a number of stages as well, for instance. And for now, I just want you to remember these four stages by name and not, and then as we move on step by step, we're going to appreciate what happens in each of them. So for now, you know what happens in G1, you know what happens in S, and in G2, you know there's a G naught phase or a G0 phase. And you know when one centrioles replicate, the cell is ready to enter the mitosis. Mitosis has four phases: a prophase, a metaphase, an anaphase, and a telophase. Now, I don't know what German or Latin they've decided they wanted to name these for, and I honestly don't know what their meanings are, but that's how you got to remember it. So make a mnemonic out of it, make whatever you can, but it's not that tough. It's just a prophase, metaphase, anaphase, telophase, and you're good to go. Okay, and as soon as it ends its telophase, because it's a cycle, it's going to enter cytokinesis, which is division of the cytoplasm of the cell equally into two. Okay, so as simple as that. So again, just to show you guys what happens in DNA replication, just so you remember, chromosome forms from these chromatids. So now we are at mitosis. Before going into individual steps, let me just highlight what I mean when I'm talking about these chromosomes. So follow my red pointer over here. Here it was a random cell in its G1 phase. If you're able, just catch my words, G1 phase. Why do I say that? Because look at these single chromatids. And then DNA replication took place. So if I draw a circle here and I ask you what processes have, what phase is in this circle, it's the S phase. So this is the G1 phase. In the S phase, it went into this state. Now this state is the G2 phase. And then as soon as it formed two centrioles, let me draw the centrioles here. It formed a pair of centrioles. So one centriole here at a right angle and another centriole here. Now these centrioles are smart enough. What they do is the second one of them is going to move to this pole and the other one will move to this pole of the cell. So what ends up happening is one of them is at this pole of the cell and the other one is at this pole of the cell. And these are the microtubule organizing centers. So what they do is they call in all the microtubules. And microtubules, I told you, are made up of alpha and beta protein subunits. So remember that line in G2 where the cell was making a lot of protein? It was the microtubule, it was accumulating. And so all of these microtubules, look at what it's doing. It's using these microtubules as ropes to align these chromosomes in the center of the cell. Look at this. It has aligned the chromosomes. Then eventually what it's just doing is it's all of it, it's going to pull the rope. For instance, if this is one of the chromosome, I attach one end of the rope here and one end of the rope here, and this is the centrosome, and I put in so much pressure that this centrosome cracks, and this end moves towards this centriole, and this end moves towards this one, and then we have the cell. Exactly, look at this one. The cell is now having half of its DNA content over here and the other half over here, exactly divided. So can you appreciate how this is happening? And then eventually cytokinesis takes place. Cell membrane pinches inward to push all of the cell contents apart and divides into two separate daughter cells that are exactly identical to each other. Is that clear?
Yes.
And
Yes.
right
Yes.
now, these are not the details. These are just the bases. Because all of you guys, when I asked you what you don't understand about this topic, you understand the details, but you've crammed those up. You've not actually understood them. So at this point, I want you to understand what's actually happening. So, right, is it a little bit okay? So, Raid's not here. He's gonna watch the video. Rima, is this clear for you as well? Do you understand how it's happening?
Yes.
Okay, that's great. So all that was happening, centriole replication, centrioles move to separate poles, they send in their ropes of microtubules and put in so much effort that these chromosomes separate. And when these separate, they just pull them together towards themselves. Cytokinesis is so smart, it's just pinching in the center just to make sure everything's divided half and half, and we're good to go. We have mitosis. Okay,
Teacher, I
yeah.
just have one question. You're saying that these are centrioles, but aren't they chromatids?
Sorry, I'm talking about these. Okay, so what I'm talking about are these centrioles. Do you remember centrioles from topic one?
Yes, teacher.
So what were
They're
centrioles?
they're the transporting proteins. They help move the things. Yeah, that's
Okay,
really.
so let me just clear your concept here. So centrioles aren't, so centrioles are just these two blocks of microtubule. They were made up of microtubule, you're right. Microtubules were those transporting proteins. Centrioles were like their task center. They were just a room sitting right next to the nucleus. If you remember, if this is the nucleus, they were sitting right next to the nucleus at a right angle, just sitting there doing nothing, indirectly controlling the microtubules. Centrioles become active in G2 phase where they suddenly replicate, and that those replicated centrioles move to opposite sides of the nucleus. If this was the nucleus, they move to opposite ends of the nucleus. Now all of those microtubules that were first randomly moving here and there in the cell to perform their functions of transporting stuff, pinching stuff out, exo and endocytosis, now they control these microtubules. What they do is, look at this here. If this is the centriole, what they're doing is they're sending in long chains of microtubules to attached to these chromosomes. This one is
Yeah, got it.
attaching to this side of the chromosome, and this one is attaching to this side of the chromosome. Now, when imagine you were sitting here and your sister is sitting here, and you're both pulling apart something from pulling apart this X-shaped figure. You have ropes attached over here, and your sister has ropes attached over here. When you pull hard enough, will this separate in the center? It will, right?
Yeah.
And when it separates in the center, this end is going to move towards you and this one's going to move towards you. So the
Okay,
basic
got it.
person that pulling these microtubules were you and your sister were centrioles. Do you get what I mean?
Yes, but in the middle, the green and the blue things, these are the chromatids, right?
They're the
Chromatin.
chromosomes. What were chromatin? Chromatin were not packed this highly. They were just
Okay.
DNA folded upon histones.
Okay.
Many of those DNA and histone complexes form something called a chromatid. These are chromatids. When these chromatids replicate, they form these butterfly-shaped chromosomes. And in the center, the ones that align in the center are the chromosomes. The ones that align on the poles are the centrioles. Do you understand it now?
Yes, thank you.
Perfect. Any more questions? Is this absolutely clear?
Yeah.
Yes, ma'am.
Yes.
Perfect. No confusion between centrioles, microtubules, chromatids, chromosomes, and chromatins. If there is any confusion, we can go back because this is the basis for what comes forward. So just make sure you know these. Okay.
Yeah.
Okay. Let's do one more slide and then we can call it a day because I don't want to overwhelm you guys today. So now we're going to the 4 phases that we talked about, or the 5 phases that the 4 phases that we talked about. And there's an extra one which we call a pre-prophase. I'm just going to tell you what that is, but let's see over here. So what's happening here is, look at this cell. It says interphase. Do you remember what interphase was? It was a generic term for G1S and G2 phase. And also, before we move on, just to point it out here, can you appreciate the lengths of these vary? Like G2 is long, S is shorter, G2 is long, all of this is short, so short, and this is even shorter. These are the ideal lengths because it takes years in G1, a few days in S, years in G2, and then mitosis takes, let's say, 15 minutes. Cytokinesis takes like 5 minutes. So this cycle, not an all accurate representation, albeit, but it is nearly close to what the actual cycle timelines look like, okay? So interphase, again, we remember what it was. We can see that. So again, like I was telling you, this DNA cannot be seen properly. Why? Because it is not yet folded upon itself. It's probably in chromatin form or DNA form. And look at this one here, centrioles. Can you appreciate this one pair of centriole right here? Now, by the end of the interphase, what we're going to have are 2 centrioles and highly condensed chromosomes, right? By the end of G2, right after G2, we enter the prophase. In the prophase, what happens is these chromosomes that were randomly here in the centrioles move to the opposite ends, and the chromosome pairs are accentuated. They are highly condensed and formed into their perfect form. And any additional material that needs to be made is now made. Any ATP or energy required by the cell is now acquired. Then occurs